Good morning, everyone. This is another talk from me about methods, but this time no ChatGPT. So the talk is called Tumultuous Fate of Sequence Bioinformatics Ideas. Instead of talking about the future, I'll talk about the past and some other things you will see. I will not redo the introduction I did yesterday. You've seen my talk in a plenary session, so this is a shorter slide. That's still my group. And so the plan of the talk today is to talk about two parts. One is sort of a behind-the-scenes point of view on a particular method called minimizer space the Brewing graphs. And another part will be about the history of sequence alignment, and especially talk about the WFA algorithm. And of course, I'm happy to have it interactively, so please feel free to interrupt me at any time. So I really liked during the retreat how Leila was talking about how she came up with projects, in particular for her research. So in this talk, it will be along the same lines. I'll give some examples of how research ideas arrive, get stalled for a while, and some might get published, maybe sometime later. You wanted to? Okay, go for it. You can take the picture if you want. So this third part will be about a work that is called Minimizer Space the Brewing graph. It was presented at Recon 2 years ago. It was published in Cell Systems. This is in joint work with Barry Chikhi and Bonnie Berger at MIT, and I'll explain what it's about. The context is long-read genome assembly. So there is now many types of sequencing data. You remember the short reads, and now we have long reads, which arguably, as you can see from my perspective, I'm biased, but I prefer the long reads. What they do now is we are able to sequence long reads of length to 10. There's some missing notation here. It's between 10 to a megabases of length. And Oxford Nanopore and PacBio's CLR technology typically had and still have high error rate. And the error rate has been decreasing, but now it's closer to 5%, which is still high for processing. And especially in comparison to the Hi-Fi PacBio, that's the one I was demonstrating yesterday for the mapping task. They are relatively shorter, but they are way more accurate, much below 1% error rate, so they achieve the same error rate as Illumina reads. So we get almost the best of both worlds, except that it's still more costly and more challenging to obtain Hi-Fi reads. But hopefully, this might change in the future. So in this talk, I will focus specifically on PacBio Hi-Fi read. Just as a reminder, the task of genome assembly consists of going from the sequencing data to getting an approximation of the genome, but you typically don't assume that you know the genome. So what you obtain is an assembly, which is a contiguous representation of the genome, which is just an hypothesis. It's not complete, it's not totally contiguous, but it gets there.
Is the 1% error rate a per base error, and is it a function of whether it's an end-del error or a point mutation?
It's per base, and it's actually way less than 1%. I was a bit conservative here. It's sometimes around 0.1% on average. And whether it consists of indels or substitutions, I don't have the figures on top of my head, but there's still a mix of the two. So in terms of assembly, you probably might have seen this in bioinformatics class. There is the classical paradigm of assembly using k-mers and the Brewing graphs, where you have the reference genome, you don't know it, but you have reads that covers the genome, and you decompose those reads into k-mers. So you remember Julia's talk this morning, this is the same decomposition. And then what is done in assembly is a graph is constructed from both k-mers, and an assembly is determined by traversing this graph. So that's what I call base space representation, because the k-mers are over the alphabet of bases, so ACTG. That's a classical, the classical formulation, and I will deviate from this just now. So you might have seen this concept before or not, so I would be curious to see How many of you have seen this? The concept of minimizers. Only a handful of hands in the audience, so I'll go ahead with it. So the concept of minimizer, it's divided into two kind of notions, which are grouped into two terms, window and universe. So window is, when you have a sequence, you consider a minimizer the smallest Elmer inside this window. So here that is the sequence. My window is an orange box. And I go over all the Elmers. Here in this example, L is equal to 2. And I consider smallest in terms of alphabet, of terms of lexicographical ordering. So A is smaller than C, that is smaller than G, that is smaller than T. So if you consider all the Elmers inside this window, the smallest happens to be the first one. It's AA. All right? So if you shift the window by one base, AA is no longer inside this window, and you don't see AA anymore in the next window. So the next smallest Elmer inside this window is AC. And you can keep going, shifting the window by one base, and you obtain a collection of minimizers across this sequence. And you can see that the property here is that consecutive windows tend to share the same minimizer. There are some considerations about ties. For instance, what if you see AC twice inside the window? Then you might take the leftmost one, for instance, to break ties. So that's one way to kind of extract a signature of a sequence. Another way is to use what I consider universe minimizers. Here there are no windows anymore. Instead, you consider a set of Elmers that you want to look for inside your sequence. And the way you construct this set is that you consider all possible Elmers, and then you assign them a certain value, and you take those which have been assigned the smallest values. So through a hash function, for instance. So concretely, once you have fixed a set of what you call universe minimizers, for instance, this set only contains 3 elements in this example, you just scan your sequence, and every time you see that Elmer, it's retained. So this is another way to extract minimizers. They're called minimizers here because they correspond to low hash values. And from now on in this talk, I will consider universe minimizers, but it can also apply to window minimizers. In the literature, there has been many names for this concept, also called frag min hash, scale min hash. It's related to locally sensitive hashing of sequences, but so the same objects have been seen in many different angles in the literature. I will call them minimizers. All right, so far? So our approach was to change the alphabet. Instead of considering ACTG, we consider that letters are the minimizers themselves. So in a classical K-mer world, you consider the alphabet to be of 4 letters. So with k equal to 3, a K-mer can be, for instance, AGT. But now, if we fix alphabet sigma to the power of L as a bit of an abuse of notation, we consider that it is all minimizers of length L. And we can enumerate them. So M1, M2, M3, and so on. So in this example, where I take L equal to 2, let's say that M1 is AA, M2 AC, M3 AG, and M4 AT. Then a K-mer over this particular alphabet is a succession of minimizers. So when k equal to 3, M1, M3, M2 is a K-mer. So it's a bit unintuitive, perhaps, because you don't see bases anymore. The bases are hidden inside the minimizers themselves. And you will see in a minute why we do this. And just for avoiding confusion, we don't call it K-mer anymore, even though it is a K-mer. We call it K-min-mer. Just to avoid saying K-mer over the minimizer alphabet or K-mer over the DNA alphabet. So once you have this point of view, you can create minimizer de Brewing graphs. Minimizer space de Brewing graphs. You take reads, you extract minimizers from the reads. So here there are universe minimizers in the same example as before. And now you can create K-min-mers from the reads directly. So for instance, one K-min-mer here is M2, M3, M2. That corresponds to the first read. Another K-min-mer here. And then you can construct the Brewing graphs over those K-min-mers using the same rule as before in classical de Brewing graphs. It is whenever 2 K-min-mers overlap by k-1 here are not bases, but k-1 minimizers, you create an edge. Question?
What happens when the length of the reads is not divisible by the number of the size of the k?
Right, so this only works for long reads. And there's no question of whether the length of the read can be divided by k or L.
Here the k is 3 for k minimers.
Yeah.
And if the read size was suppose 10, then how it will account for the last base, the 10th one, because 3 is not.
Oh, yeah. So what's happening here is that there's a lot of loss of information. Like the bases in the reads, you discard actually a lot of them. Like for instance, this T here, it's not part of any of the minimizers I've extracted. So it's as if it didn't exist. So some of you might have noticed that this transformation from reads to k-minimers is extremely lossy. It's not lossless at all. And in principle, if you think about it, when you transform reads to k-mers, it's also lossy because you lose the order. And that was in fact a question during the first talk this morning. So here, not only you lose the order, but you also lose bases. But hey, you get something else in return. I will talk about it later. So then the formal definition is a minimizer space de Brewing graph is the Brewing graph over the minimizer alphabet. That's it. There's no more to it. So once we develop this, we apply this concept to genome assembly of hi-fi reads. So the representation of the pipeline is as follows. You have reads, you convert them into minimizer space. Then you construct the mDBG over the k-minimers. You do the assembly steps that people typically do in assembly graphs to remove errors, to remove variations. And then you go back to base space. You retrieve the base sequence for each k-minimer of the graph, and that gives you the assembly. So that's a simple enough pipeline. And we applied it to the assembly of PacBio reads. And the highlight here is that it's very quick, because depending on how you set your parameters, k and L, you discard a lot of bases. So 99.9% of the bases are discarded, but you can actually retrieve them in the last step. But all of these steps are now very efficient. So we could do de novo assembly of human genomes in 10 minutes and 10 gigabytes of RAM, whereas the other tools are way more expensive. The quality is okay, but not perfect. It's very far from telomere to telomere high quality assembly, so you get a draft out of it.
Could I ask a question?
Sure.
So, genome fraction is the way that you're quantifying accuracy?
Yeah, no, it's completeness. So if you know the QAST assembly evaluation tool that is used to evaluate the quality of assemblies, we run it, and this is the metrics that it gave us. So NG50 is related to the fragmentation of the assembly, the contiguity, and genome fraction is completeness. So how much of the original genome we should have recovered.
So, like, do you know just off the top of your head if there's an indel, like an indel of 100, like how would that be quantified in the genome fraction?
It wouldn't be reflected by this metric. Like, the genome fraction really is more macroscopic than this. The base level accuracy of the assembly is reflected by another metric that I didn't show here. But the base level accuracy of the assembly here in our pipeline is going to reflect the error rate of the reads, which is like 0.1% to 1%. We don't claim to have a good base level accuracy at this point. It could be polished later, though. So this is really a draft assembly. Think of it like this. You get a quick and dirty, fast assembly of a human genome using this method. All right. We also applied this to pangenomics. So we took 500,000 bacterial genomes that were available and assembled previously by Blackwell et al. It's a very big file. It's a 3 terabytes file. I'm not going to do any demonstration over this file. It's unmanageable. Even with the tool, mDBG, that constructs this Brewing graph, we run it with those parameters, and it gave us a huge result, a graph that is so big that it is impossible to draw it. The only thing I was able to do is extract the 5 largest connected components and plot them using bandage. So those connected components correspond to minimizer space de Brewing graphs of some bacterial genomes. And here I looked at what were the dominant species. So for instance, if you look at this circular connected component, it's nice because the genome of Salmonella enterica is circular. So you do expect to see this shape. And essentially, what you see here is all the sequence variation of the pangenome of Salmonella, summarized into an mDBG. And there are 700,000 more connected components of other species in this graph. So it's extremely difficult to analyze, but at least we could construct it. Before, there was no pangenome graph for this collection. Just for fun, we also plotted the same graph, but for equalized genomes. So we took all of the existing equalized assemblies and made an mDBG out of it. And it's a bit surprising here, because if you know something about bacterial pangenomes, the difference between two bacterial genomes is often very large. They don't share a lot of sequence. Yet, here we almost discern a linear path in this graph, but we are not supposed to, because the pangenome of E. coli is not linear at all. It consists of many regions which are not shared across genomes. So again, that's a case where we could construct the graph, but the analysis of it is extremely challenging. All right, so this was basically a talk I could give at a conference about mDBG, giving you what are the methods and the results, but now I want to spend a few minutes explaining behind the scenes of this project, because if you only heard what I said, it would look like a standard project, but at the beginning, we did the work, it ended. But it wasn't that. So, okay, what I also talk about in conferences sometimes is this slide. It explains how we came up with the idea. Essentially, there are 3 different methods before us, Shasta, Peregrine, and WT-DBG2, that each had some parts of that idea. So for instance, in Shasta, they had the idea of transforming reads into lists of minimizers, but they didn't consider k-mers. In Peregrine, they had the idea of indexing pairs of minimizers, but they didn't do the Brewing graphs. And in WT-DBG2, they did the Brewing graphs. They did a change of alphabet, but they didn't consider minimizers. So if you know these 3 works, and if you think hard for a long time, you can come up with mDBG. However, the behind the scenes of the project, I think, can be interesting. So in summer 19, the first prototype was developed, but it just didn't work. We couldn't assemble nanopore data. That was the goal at the time, because nanopore data has too high error rate. It worked very fine for error-free reads, but that's a bit useless. So the project was dropped for a year, like no more development. And then we picked it up again. We focused on developing an error correction algorithm so that potentially it could work on assembling nanopore data, but we failed. We could not develop a satisfactory error correction algorithm for mDBG. So then we decided to pivot and just focus on assembling hi-fi reads. And then it worked well, because hi-fi had a very low error rate. So we published immediately a few months later, which was very timely because many other competing works appeared at the same year. So mBG, the T2T assemblies, LGA assemblers, all of them also focus on assembling reads, hi-fi reads. So what I like to show also is that the very first prototype of mDBG was just 100 lines of Python. And it was enough to determine that the IDE could work for some use cases and couldn't work for others. So if you remember my talk from yesterday, you know that my coding style is not always the best. So this is my, you can basically see what the steps are. This is my FASTA parser in 10 lines of Python. This is a way to extract minimizers in like 10 lines of Python as well. We constructed a basic network X graph, so we used the library for it. Then we opened the reads. You can see which reads I tested. We constructed the minimizers. We created the nodes of the graph here, created the edges of the graph here, and we saved the graph, and that's it. So that was a prototype, and it was like nothing sophisticated at all, but still sufficient to develop the idea. So I guess my takeaways from this part is that projects can be stuck. You can pivot ideas to shift the project to a different focus. And the big leaps should happen quick because the field actually moves fast, and that project, if we waited 1 more year, it might not have been worth developing further. In terms of the future of the mDBG ID, we realize now that it doesn't provide high-quality human genome assemblies, so that wouldn't be the right focus for this project. However, it seems to work well for pangenomics, and in fact, we also applied it for, this time, a well-performing metagenome assembler that we pre-printed a few weeks ago. And it is also the basis of MapQuick that I demonstrated yesterday for alignment. So now I would like to give some history of the concepts behind what I talked about earlier. So the concept of minimizer, we didn't come up with it. It actually was, to the best of my knowledge, first introduced in '95 by Cenk and Zainab and Vishken. And it took 17 years for it to be applied to sequencing data. So that idea was dormant for 17 years, even though sequencing data appeared, well, in the 2000s. And when it was first used, it became very successful in many other tools, including some I developed. The concept of the Brewing graphs is even older. So if you really want to be mathematic about it, you can argue that it was developed in 1894 by Camille Fly Sainte-Marie. And the article is difficult to come by, but it's actually really talking about the Brewing graphs in the previous mathematical world. And it took 100 years for it to be applied to sequencing data, but this is because sequencing data didn't exist 100 years ago. But then still, between the first conceptualization of it to the first assemblers that used the Brewing graphs, you can see that, 10 years occurred. On the terms of optimization of the space of the Brewing graphs, it also took many years. So from, let's say, 2001 to 2009, there was no consideration of optimizing the space. But now there are many, many tools that aim at optimizing the Brewing graph data structures. And I talk about them in an article here. Well, mDBG is way younger, so there's for now no history of it. Any questions on this part before I move to part 2? If not, I'll move to part 2. I want to briefly talk about WFA. Who is aware of what it is? No one. OK, so this is going to be an alignment novelty for many of you. So the best way for me to explain WFA is just to quote and read this tweet. And maybe explain a little bit what it says. So WFA is an alignment algorithm, similar to Needleman-Wunsch, which you might have seen in your bioinformatics courses, which is the most famous alignment algorithm. And the difference here is that it has a different penalty for gaps. Instead of a linear penalty, it's an affine one. And the novelty in terms of performance is that its time is linear in the divergence of the sequences instead of quadratic in the length of the sequences. So it's much faster. And Hangley considers it a breakthrough, and I would totally agree. It was published 3 years ago now. And I'll explain a little bit what it's about. So just a quick recap. So classical sequence alignment looks a bit like this. It's dynamic programming algorithm. So you put the two sequences that you want to align to each other as columns and rows. So this is sequence 1, this is sequence 2. And you fill in a matrix of scores using a certain rule. And essentially, you try to determine what is the score at the bottom right of the matrix, which gives you the alignment score. And the rules that are used to fill in this matrix are simple. You consider the neighbors of a bottom right of a certain cell, you consider the top neighbor, the left neighbor, and the diagonal one. And you essentially apply a max between those three expressions. And those three correspond to the value of the previous cells plus a certain penalty. And you have to set the penalty a certain way. And then it's an automatic way to just fill in this matrix. And at the end, you obtain the alignment score. You can do global alignment, as I explained here, or local alignment to just find some local similarities. It follows a similar spirit. So that's Needleman-Wunsch. For the local version, it's called Smith-Waterman. And here with WFA, it follows a similar approach, but different in a crucial way. The difference is that it considers that when you have identical substrings, for instance, AAC here, you can see that they are identical between the two strings, it immediately skips filling scores in a diagonal and directly puts the right score on at the bottom, at the end of the identity of strings. So this optimization allows to fill in the matrix much faster by focusing on diagonals instead of focusing on all cells of the matrix. And you can kind of see now that with this strategy, the complexity of the algorithm will not be no longer dependent on the length of the two sequences, but on the similarity of the two sequences. So explaining properly WFA would require a proper complete talk, in fact, at CGSI, so I will skip the details, but hopefully you understand the intuition. So as I said earlier, the benefits now that the running time, it depends on N and S, so sequence length and divergence. And S is typically small for genomic sequences. So it's in particular way smaller than N. So this gives a big benefit compared to alignment in O of N squared, which is in the range. So it's defined for certain gap penalties that are useful for aligning genomic sequences, and the space is also small. So S squared, which has been further optimized to O of S in later works. And what is really interesting for WFA is the history of this algorithm. So since 1985, 86, there has been no method that could extend alignment algorithms to affine gap penalty in O of NS time. So it took 3 decades, as said Hangley, for an algorithm to be found. Even though sequence alignment is perhaps one of the most fundamental problems in sequence bioinformatics, still took 30 years for this algorithm to be discovered. On the space front, there has been also developments, and aligning sequences in small space is crucial for being able to align long sequences, for instance, complete genomes. What I will briefly mention now is the state of the art of aligners. So there are many tools. One of the most famous one, I would suppose, is KSW, which implements a very optimized version of Smith-Waterman using SIMD. And basically, I took all of these tools and compared them in terms of speed. And the results are quite surprising. So on the x-axis of this plot, I give the divergence between the two sequences that I'm aligning. So I created a benchmark with many different divergences. And on the y-axis, there is the speed of alignment in terms of megabytes per second, to just normalize it. And you can see that WFA is very fast when the sequences are close to identical. Here, it's 1% divergence. However, as soon as you increase the divergence, predictably, it becomes slow. The other tools that perform relatively consistently are variations of Smith-Waterman, which are highly optimized. So this informs the use of these algorithms. So when you have, let's say, PacBio hi-fi data, then WFA is very nice. But if you have genomes which have high divergence, then it would be advisable not to use it, because then you would fall into this regime. What is interesting is that KSW2, that is used in Minimap2, you can't even see it in this plot. It's like close to 0. So that shows a big benefit for a big potential benefit of incorporating one of these recent alignment algorithms inside popular alignment tools. So one may ask, why is WFA not incorporated inside Minimap2? And the answer is hidden inside a tweet thread from 2020, where Hang explains that since WFA cannot extend indefinitely alignment, it needs to do global alignment. There is a notion of G-local that is possible but hasn't been developed yet. And the author of WFA says, yeah, actually, you could actually perhaps do it. And then they start developing the idea and they stop here. So 3 years ago, there is like a beginning of a project that you can see on Twitter, and the project never came to life. That's very interesting, I think. In terms of being able to map fast without alignment, we did a benchmark this year, and we benchmarked Minimap2 with many other tools, including MapQuick, which you saw yesterday. And basically, we show that there is already a potential to do mapping very quickly. And for instance, as you saw, MapQuick was like 30 times faster than Minimap, and even way faster on more complex genome. Here it's maze. So there is definitely ways to improve the state of both mapping, so finding coordinates of reads, and alignment through WFA, for instance. I will just list some open problems if you are interested in working in this area. So there's clearly the question of developing WFA extension algorithm, but nobody has picked up yet. There's also clearly the question of optimizing the implementation of WFA using SIMD so that it's faster across all divergence levels. HMM alignment also hasn't been really touched by the community since a long time, which is hidden Markov models. The state of the art has perhaps a bit stagnated compared to other alignment procedures. So this might be another case where we are in a 30-year of no research area. An increasing sensitivity of k-mer search would also be very desirable. I have no idea how, but it would be nice to do it. Okay, so that was my last slide. Thank you for your attention. Happy to take any questions. Yeah. Oh, the last one. So k-mer search or k-mer alignment, you know about space seeds. It's a way to remove some nucleotides inside k-mer to allow them to match anything. So this increased sensitivity because you are no longer constrained to mapping exactly the sequences. The main drawback of using k-mers is that sensitivity is typically low compared to like BLAST or other alignment tools. It would be very nice if we could find a way to use k-mers for speed and at the same time have sensitive alignment. There are potentially ways to do this, but it hasn't really been explored. Yeah.
Questions. So you kind of distinguish mapping from alignment.
Yes.
I was just wondering, I mean, because the way at least I think of it is often you align to be able to figure out the reader's mapping in a given position. So I'm just kind of trying to understand what the distinction is and why that distinction is important.
I don't think it is a very established vocabulary in the community, but I like to call mapping just finding the coordinates, the interval of a sequence on another one. And alignment is really the base, pair base correspondence of the content of that sequence versus the reference. So think of it as a difference between a SAM file with cigar string and a SAM file without cigar string. So cigar string essentially tells you where are the indels, where are the SNPs. And you can also construct a SAM file without a cigar string, which just tells you this read, it maps here. But I'm not telling you how it maps. So I prefer to make this distinction because when you run Minimap2 with default parameters, what you get is mapping. If you add the -a parameter, you get an alignment. So there's a way to do both, and getting alignment is actually much more challenging. Mapping is very quick, as we show here. We can map a human genome in a minute. However, we cannot align reads to a human genome in a minute. Yeah.
Follow-up question, I mean, different question. Like how useful do you think are compute architectures like GPUs in speeding up this, and what's the state of the art?
There has been some developments in adapting some of these tools on GPUs, but two things. So the speedups haven't been stellar, and the adoption of GPUs is not high in terms of bioinformatics clusters yet. So there's potential. I know that NVIDIA has created a suite of tools, including Minimap2 on GPU. So it's faster, but it's not orders of magnitude faster. It's a constant that is sometimes somewhere between 2 and 5. So I think there's a big potential to developing novel algorithms instead of speeding up existing ones using different architectures, especially given the data that is evolving.